Recording in progress.
Awesome. Okay, so yeah, fairly straightforward. Let's dive right into this handout. I hope our friends at home are also looking at the same handout over here. Correct, right? Same, right? Everybody looking at the same one, huh? Awesome. Thank you. I appreciate the head nods. Okay, so before we actually look at the questions, the 4 topics that I have chosen to include over here, yes, they are in sequence, right? Can we just have a look at the 4 topics here and think about which is the not the most important, but the most recurring topic we have over here? Which of these 4 topics is the most recurring? I actually kind of heard an answer. Who muttered that? If you don't mind sharing. Okay, let me just try and remember some names then. No, yes, Enqi, was it Enqi? Who's Enqi? Oh, hi, Enqi. Which topic do you think is the most recurring? Movement of substances. Can you share a little bit about why?
Diffusion, osmosis, and electron transport.
If I could give you a gold star, I will. Because yes, that's exactly what I'm looking for. Movement of substances, right? It is a very recurring topic, right? You're partially correct as well, right? It is something that appears many times, right? Specifically, the explanations of how those processes occur, right? In our pure bio syllabus, we're learning the 3 processes. Lim Zhen, can you help to? Do I call you Lim Zhen? Okay, right. What are the 3 processes we learn in movement of substances? Transport, thank you. All right, so diffusion, osmosis, and active transport. So they are very recurring because exactly like what Enqi said, they appear in many of our future topics. All right, okay, and that's what we will actually see in the second session as well. We look at nutrition, we look at transport as well as respiration, and even excretion as well. Okay, so without further ado, this is a 2 hours. We're just going to focus on the worksheet. We're going to do it together. We're going to dig deep and try to recall all these little nooks and crannies of the concepts and try and get as unblur as possible about them. All right, so first question, learning point here is to actually be able to identify from drawings and diagrams. So over here, I have given to you a drawing. It is very reasonable to expect you to be able to identify cell parts from an actual photo micrograph as well, right? So let's say a researcher looks at a cell with a light microscope. They take a picture of that. You need to be able to identify that as well. Okay, so over here, first step is to actually identify what these structures are. I always tell myself, whenever I see a diagram that is labeled, no identities, no names, that is the first thing I do. Can I offer us 30 quick seconds to try and identify these 4 structures, please? Okay, please help me with this. Sarah, choose one letter and identify it for me, please. Where is Sarah? Sorry. Oh, yes. Hi, Sarah. Hey, give me a while to remember your names and everything. Okay, Sarah. Hey, don't just sit down there. Wait for answer. Okay. Wait, Sarah, I give you a chance. I ask someone else first. Later I ask you again. uh, Umaira, choose a structure for me, please. Y is the nucleus. Very good. Choose the most difficult one. Wonderful. Nucleus. Okay. Sarah, ready? Which one do you want to identify for us? Z is the Golgi body. Wonderful. otherwise known as the Golgi apparatus as well. Okay, Kiza. Kiza? The? K. Oh my goodness, sorry. Kiza. Come, Kiza. Choose one letter and identify for me, please. Okay, X is the rough ER, is it? Okay. And finally, who is the lucky one? Let's look for 1K. Hi. Okay. W is the how? Let's have someone help us. Anika, what is W? Yes. Okay, does it make sense? Okay, so what we're looking at over here, we can see, although it's very pixelated, it is very obvious that these structures here have tiny little dots here, while this segment, no dots. Correct now. Okay, so rough ER and smooth ER. All right, so here we are looking at recalling structure, pairing structure to function, right? A very important skill for us to have that brings us through our entire bio journey, being able to identify structures and pairing it to their functions. Okay, so I will first tell you X is incorrect. I would like to hear from you why is statement one is incorrect. So why is that statement incorrect? Hui, can you help us with this, please?
What is the word is that? Who knows?
Okay, continue first. Why is statement one incorrect? X is the site where polysaccharides are synthesized.
Could be.
Okay, good enough. Right, so this X over here is a little bit vague, but it's fine because it's an MCQ question. X can refer to the entire rough ER, or you can refer to the ribosomes. Thank you, Lipson, right? So the ribosomes as well. Regardless whether is it the RER or the ribosomes, statement one will be incorrect. Do we agree? All right, statement one will be incorrect, regardless of whether X is the ribosome or the rough ER. Okay, strictly because of this word. So Hui was almost there. Polysaccharides are not glucose. In fact, glucose is a monosaccharide. Very good. So, can I have an example of Zixing, can I have an example of a polysaccharide, please? Who is Zixing? Zixing. Hi, yes. Hi. Can I give an example? And in our syllabus, we learned three of the. Anyone, please? That is still a monosaccharide. Come, someone else, please. There's the one in the water. Emily, who is Emily? Where is Emily? Hi, Emily. Can I have an example of a polysaccharide, please? Starch, awesome. You sound confident. Do we have the other two as well? Awesome. Good stuff for you as well, huh? Okay, so these are our three polysaccharides: starch, glycogen, and cellulose. Okay, these are the carbohydrates. Polysaccharides are carbohydrates. So essentially, when we think back to the function of the ribosomes, it is about protein synthesis, not carbohydrates at all. Okay, right? So here, that's why statement one is going to be incorrect already. Okay, Y controls all cellular activities in the cell. Absolutely correct. That's what the nucleus does. Okay, Z modifies the substances made in Y. Okay, Sarah identified the Golgi body to be Z. And if I were to assign one word meaning to the function of the Golgi body, it will be modified. But why is statement 3 incorrect? Again, sorry?
It's not me.
Very good. The made in Y portion is the one that makes this sentence incorrect, right? Z modifies substances, yes, but generally they modify proteins. Okay, in your syllabus, we're just going to keep it simple. The Golgi body modifies proteins that have come from the rough ER, not in the nucleus. Can see that? Okay, so that is why statement 3 is incorrect. And so we are definitely left with 2 and 4. So 4 is correct. W carries out detoxification. And this is not something that we need to explain in very detail over here because your syllabus only requires you to remember that, hey, yes, the smooth ER is kind of like one particular organ in the body. The smooth ER detoxifies cell content, just like how the what detoxifies our body. Do we know the answer? Let's learn more interactively. If you have an answer, please share with me. I don't need to shout it out. It's a small classroom. Just tell me, what organ am I thinking of? Kidney. Oh yeah, kidney is important in cleaning the body, yes. But the kidney removes something from the bloodstream. This other organ removes many, many things. The liver, we're looking for the liver. Right, thank you, Lim Chen and Hui. Right, so we're looking at the liver. So the smooth ER is like the liver. Okay, it cleans the cell content. Okay, so we are looking at 2 and 4. Answer is C. All right, we have a couple of questions from our friends at home. Okay, let's see. How do we know if the arrow is pointing at the nucleus or the nuclear envelope? Very good question. At this point in time, for this particular question, makes no difference. Okay, that makes no difference because okay, your name put letter N only, so I can only call you N. Okay, because N. Alright, at this point, we are looking at functions of the cell parts. It wouldn't really make sense for me to think about what's the function of the nuclear envelope. Keep the genetic material in. If that's the case, then I cannot answer my question already. Okay, we need to be more specific when it comes to open-ended questions instead. All right. Hope I have answered you there. And for Brian, same answer to you. Ribosome versus RER. For this particular question, doesn't really matter, right? So you look at the options given to us. See what the examiner is trying to pull out from you, whether you know the function of the ribosome or the function of the rough ER. All right, Brian, I hope I've helped you understand that a little bit better as well. Awesome, let's go on. Live pancreatic cells were extracted from a mouse, kept in an isotonic buffer solution. Now, Pure Bio, I want to dig deeper with all of you here. Isotonic means what? Clarissa, can you help us to remember what isotonic refers to? Where have you seen this word isotonic before? Maybe outside of the textbooks, the drink, right? So when we drink isotonic drinks, what is it for? Anybody thought of it before? Or just like, yeah, nice drink. Clarissa, any guesses as to what the word isotonic refers to? Sugar, what about the sugar? A lot. Yeah, you're right. Very unhealthy because it's very, very sweet. But we're not looking at sugar actually. What else? Jenny says give energy. Oh, I see the correct word. We're looking at electrolytes, and Brian has correct word as well. Concentration. To be less fancy, we don't say electrolyte. We just say ions. Okay, our isotonic drinks, we refer to them as isotonic because they have the same level of ions that the body requires. So after you exercise, after you sweat, you use up a lot of them. You lose a lot of them. You drink to replenish. That's the theory behind it. But it's always very unhealthy because, like what Clarissa mentioned, it is too much sugar already. So the cons outweigh the pros. Okay, back to this. In bio here, what are we looking at for the meaning of isotonic? I would love it if you all can write this down with me over here. We're looking at same concentration slash WP as the cell. What would WP refer to? Eating, thank you. Water potential. Okay, so this is very important for us to look at our water potential. I need to maintain cells. If I want to store live cells, I need to store them in this isotonic solution to prevent the cell from doing what? From what? To prevent what from happening to my cells? to prevent osmosis from occurring? Yeah, it's kind of that. Because if there's too much osmosis entering, what will happen to my cell? First, if there's too little, not if there's osmosis exiting now, what will happen to the cell? Crenate. Thank you. Right. I want you all to keep those in mind. Later the FRQ has. Okay. So, and then the next one we're looking at maintain at 37 degrees Celsius. Right. So that is body temperature of the mouse as well. Right. So most mammals have this similar body temperature lah. Okay. Radioactive amino acids. Right. The word radioactive does not mean that you will die. Right. They have a bad rap. We use radioactivity in a multitude of reasons. Here we are using it to follow what happens to the amino acid. Simply put, this question is asking us to recall the processes of protein synthesis. I hope we all remember that amino acids are the building blocks of proteins. So therefore, when we are following the processes of protein synthesis, where does it occur at? And then where does it go to next? And then following what happens next. Okay, so what is the thing that synthesizes proteins first? What is the thing that synthesizes proteins first? Jingxuan, what is the site of protein synthesis? Ribosome. Awesome. Hey, wow, how our options don't have ribosome. So Jingxuan, what would be our better choice next? Next better choice. The RER, very good, right? So over here we are looking at the RER is the one that contains the ribosomes, right? So we're going to start over there. So between B and C, this is our next step already, between B and C. After RER, does it go to the Golgi or does it go to the smooth ER? What is the word? What is the one word that we will assign to the function of the Golgi? I share with you all earlier on. Modifies. Thank you. Modifies. It's a right? Yes, okay. Right, so it's modified. So after being synthesized by the rough ER, at the rough ER, sorry, it will go to the Golgi to be modified. And then, because it is the pancreas, it will need to be secreted out of the cell. So therefore, our answer is C. Yes, you're right, Brian. Okay. Awesome. Right. At any point in time, if you want to ask any more questions or just clarify stuff, please feel free to let me know. Those in class here. Okay, so a very typical question as well, right? In question three, not necessarily always have to be viscine tubing. So over here, I just put it down as our partially permeable membrane. Okay, so let me help you understand this a little bit more quickly. Starch suspension is inside the tubing, and then I have potassium iodide outside the tubing. Okay, we all know this since primary school. When iodine comes into contact with starch, we observe a color change from yellowish brown to dark blue or blue-black, pending. Okay, over here, we can see that our observation is that inside the tubing, blue-black. So previously, inside only got starch. Now it turned blue-black. This can only mean this happened. Do we agree? This blue arrow shows that the iodine entered the tubing. Okay? Did the iodine outside turn blue-black as well? It didn't. So therefore, starch did not exit. So, wait, let me draw it. Okay, so it could not exit. So therefore, what is this property that we want to keep in mind here that explains, why iodine can move, starch cannot? What's the difference between iodine and starch over here? That allows only iodine to move, but starch cannot move. What's the difference between iodine and starch? Concentration. Okay, if we think about concentration, only starch here, no starch here, mean I have a gradient. Correct now. So according to the gradient, starch we want to exit. So it's not the gradient. Thanks for trying. What else? Sorry? Suspension versus solution. Facts, yes. Not relevant to our explanation here. Thanks for trying. Yes, our friends at home have got it already. What's the difference between starch and iodine? Trying to find some things. 1K, come you again. What's the difference between starch and iodine? Yes, well done, Jacinda, Athena, and Brandon. It's very straightforward, right? Yeah, starch is humongous. What is starch? It's a polysaccharide, isn't it? We just talked about it. Polysaccharides means they are made of many glucose molecules. Iodine is I2. Glucose is what? Do we know the chemical formula of glucose? C6H12O6. Starch is many of those. Iodine is just I2. Right? So if that helps you understand a little bit why there's a difference, starch molecules are larger. Starch molecules are larger than the pores of the membrane. Okay, so iodine can pass through the pores of the membrane. Starch cannot. Just like how an elephant will not be able to enter the door here. Okay, so just keep that in mind. Alright, so over here, if we see these two options, we definitely don't choose C and D already. Because C and D talks about removal of starch. Okay, between A and B, which one? Hui is the obviously wrong one between A and B.
A.
A is
Yeah.
yes, B is the wrong one. Why?
No, the molecules.
Very good. Osmosis is specific to water, and iodine can't be osmosis. And our answer is A. Okay. Okay, moving on. Food tests. Okay, so we just came from potassium iodide. Oh, this is iodide. Sorry, it's not iodine. It's supposed to be iodide. Please make the change for me. Iodide. Okay, this is a test for. Okay, this one very simple. Blue black is a positive test. Means got starch. Okay. Now, Umaira, help me out here, please. Benedict solution is a test for. Benedict solution is a test for. Ooh, too specific. No. It's a test for 5 things. Glucose is one of them. Starch is carbohydrate. Sarah, do you have an answer? RS. 2 words. Yes, thank you. Reducing sugars, right? Reducing sugars. And Yumara, still you then. Which one is a positive test? Brick red or blue? Okay, if I give you a clue, Benedict solution, right, originally contains copper 2 ions. So therefore, red is the color change, right? Then you can use that to remember that as well. Okay, awesome. Thank you. All right, so over here, Brandon, hold on. Wow, a good question. Finally, what's this? I never learned this thing. I got no name one. What test is this for? Do we know this? We know that. Do you know what are we testing for with this? Sorry? Is it ethanol emulsion? In the ethanol emulsion test, I have to add ethanol. Correct or not? This one not ethanol, ma. Okay, so what is it test for? We only learn 4. So if it's not that, then the last one is protein. What's the name of that test? Puret's test. Wonderful, ladies. Thank you. Let's write it down together. Anika, violet or blue is positive. Use the same, remember the same question I asked Umaira? Violet. Okay, see. Copper 2 here, right? Chemistry QA. Copper 2, what color? Blue. So therefore, this one means I added blue. Cannot be this one is a positive test, right? I want to observe color changes, right? Okay. So I have identified for you which one is the positive test, which is the negative test. Go ahead and look at our 4 options here. Which one is the correct option? Uh, Brian, I hope I've answered your question. Jenny, the idea is in your head, but wrong spelling. To make sure you get that right. What else? What else? Wonderful question, Kaden. Let me just finish it up first and I'll answer you up. Okay, so let's hear from Auntie. You chose B for boy, is it? Okay, starch and glucose in X. Hey, Auntie, this one means no starch already. Sarah, do you have an answer? C is the answer. Why C? What does egg white have? Protein, that's it. Okay, egg whites contain protein. So our answer is C because in tube Z, we have proteins, we have starch. I never say anything about this. That's our answer. Okay, right. Bonus question. What is the name of the protein in egg white? Anybody? It's a very common blood protein as well. Albumin. Right? Albumin is the name of protein found in egg whites. It is found in our blood as well. Okay. Good try, Brian. Right. Oh, cannot be hemoglobin. If not, the egg whites will be egg red. Okay. All right, good try. Okay, so I have a couple of questions I want to share with everybody as well. The first one is maybe I can direct this back to my reducing sugar for how many? Five, glucose. What are the others? Come, let's help our friends here. What are my reducing sugars? Galactose, maltose, fructose, lactose. Right? We need to remember we have 3 monosaccharides and 3 disaccharides in our syllabus. So from these 6, just remember sucrose is not the reducing sugar. Okay? Alright, this one go back and check your textbook, whatever notes you have. Very important. Oh, I think I have it in the revision kit. Yeah, I think I have it in the revision kit. Let me find out. I think it is at 11, page 11. Right? So those with the revision kits, you look at page 11, I have categorized it very nicely for you there. Okay? Awesome. Right. The next question that I want to share is, if you look at our Burex test, sodium hydroxide and copper 2 sulfate, is there a significant difference if I add sodium hydroxide first, then copper 2 sulfate, or the other way around? Is there a significant difference if I add in sodium hydroxide, then copper 2, or the other way around? Emily? No significant difference. Yes, that's the answer. There's no significant difference here. So why do we need 2 different reagents then? Let me tell you right now. The only one that results in color change is the copper 2. So why do I need a sodium hydroxide? Think back to, okay, very simple. Chemistry. What is sodium hydroxide? Just need one word from you. Alkali, thank you. Okay, sodium hydroxide and alkali. Now back to bio. What do alkalis do to proteins? So many blank faces. What do alkalis do to proteins? If the protein, the pH deviates too much, what will happen to the protein? To the enzyme? Denature. Denatured means it loses its shape. What we're doing here is opening up the protein so that the copper 2 ions can react with the insides of the protein more easily for the color change to occur. Okay, right? So this is if you really want to optimize your understanding of how the Buret's test works. Okay, there's a reason why it is a two-part compound, two-part mixture for the test. Okay, all right, we can dive into greater detail next time if I do see you in our classes. Yeah. But for now, let's move on. Wonderful question. I think it was Kaden, right? Okay, so question 5. Right, Sharon was eating a piece of unsweetened bread. So it is just bread. You chew, 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 chew. After a while, wow, tastes sweet, right? So this can happen to rice as well. Noodles. Anything that is made of what? Bread, rice, noodles. What's the common thing here? Starch. Thank you. Starch. So if you chew a starchy food, starch is not sweet at all. You all didn't realize starch is not sweet at all. But if you keep chewing starch, keep chewing, I thought it would taste sweet already. Why? That phenomenon is one of our answers here. Can you make a guess, please? Vincent, what do you think? Into? Yeah, that's it. So it's the answer. Okay, well done. Answer is B, right? So salivary amylase in our saliva. So Linton has helped us identify the enzyme in our saliva here. And we have broken down the starch into maltose. Now it's very important to remember the amylase only breaks down starch into maltose. Okay? Kaiser, what does maltose, what breaks down maltose? Maltase, very good. Please keep in mind, enzyme names, enzymes are always named after what they break down. So the enzyme that breaks down maltose is maltase. Okay, right? Maltose will be broken down by maltase, Anika, to form what? Maltose is broken down by maltase into glucose. Anything else? Yes, no. No, very good. All right, because maltose is made of glucose. Okay, wonderful. Thank you. Right, so yes, question 5, answer is B. Let's go. Once more, egg whites. So this tells me it's going to contain proteins. Okay, then why are we using the enzyme, the gastric enzyme? What is this gastric enzyme? What is this gastric enzyme? So a little bit of overlap with the nutrition here as well. Must remember. Yes, Jones got it right. What is the name of this enzyme in the stomach?
Recording in progress.
Someone answer me, please. Nobody? I have 13 beautiful minds here. Someone share with me the answer, please. Pepsin, thank you. Pepsin. Pepsin is what kind of an enzyme? What does it break down? Proteins, right? So this gastric enzyme over here, specifically, we are looking at pepsin, which is a protease. Back to basics. ACE tells me it's an enzyme. The words before that tells me what it breaks down, which is therefore the protein. So pepsin is a protease which breaks down proteins. And we already know that egg whites contain proteins. This is why this reaction can occur. So understanding how this works over here, the cloudiness in the test tube is due to the proteins from our egg white. After adding my gastric enzyme, I wait for 8 minutes. Oh, the cloudy appearance disappeared. It looks clear now. So this means that the proteins in egg white were broken down. Question now is not asking why that happened. Question is asking us, how can I make this disappearing act occur more quickly? How can I make this disappearing act occur more quickly? Here, are we trying to increase or decrease the rate of reaction? Are we trying to increase or decrease the rate of reaction? Increase, thank you. Okay, so with that in mind, I want to increase enzyme activity, isn't it? Increase enzyme activity. Which of my four options achieves this? Killian and Brandon got it. Well done. Zixing? Sorry? D, is it? Last one? You chose raising the temperature. Okay, very good. Raising the temperature. When I raise temperature, the enzymes and substrates have more what? Higher temperature means more what? Why do they have more collisions? Why would they form more enzyme substrate complexes? Yes, kinetic energy first. Go back to the basic points first. Okay, raising temperature, enzyme substrate, more kinetic energy, move faster, more greater frequency of kinetic collision, blah, 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 blah. Oh, that's the wrong answer. That's the wrong answer. Why? Look at that. 65 degrees Celsius. Where did I get the enzyme from? Your stomach. If I put you at 65 degrees Celsius, you will cease to be you. Okay? So enzymes that are found in a particular organism will have an optimum temperature close to that organism's body temperature. Our body temp, 37. So any of our enzymes in our body optimum temperature will be close to that. Logical? Okay, so therefore 65 is not just too high. 65 is above optimum temperature. Yes, Brenda, you're right. Okay, so 65 is above optimum temperature, so therefore D is the incorrect answer. Yes, Jenny. Right. Okay, so ABC, we're left with that. What are we going to choose? ABC. C is the correct answer. Because what is the condition in our stomach? Acidic due to hydrochloric acid. Thank you. So our answer is C over here. Okay, bringing it closer for optimum pH. Okay, awesome. Let's go. Now, question 7 is an interesting question. Can I offer us 2 quick minutes to read the 2 paragraphs there and try and understand the diagram? And hopefully choose the correct answer. Because difference here is the boiling thing, okay, because difference here is the boiling thing, what does boiling three do to the seeds? Softer. Okay, I'm not gonna eat the seed, so boiling not about cooking, so yes, those things occur, but I want to link back to a concept over here. Why am I denaturing, Vincent? The enzymes. Okay, I'm denaturing enzymes. Okay, we are on a recurring theme, okay, so we must first understand and recall what is the core idea of a seed. What's the purpose of a seed? What's the purpose of a seed? Xingquan, what's the purpose of a seed? Again, to sprout. Okay, so to germinate, right? Okay, so it will grow its root, grow its shoots. During this entire process, it's a plant, right? Can it photosynthesize yet? No, then how does it get its energy from? How does it get its food from? The shell? Is it not peanut? How do seeds in general get their food from? Where do they get their food from? This is a primary school thing. Think back, like before, many years ago. Umara, do you have an answer there? The seed itself, the seed leaves contains stored food, isn't it? That's what we use in primary school, stored food. So now let's jump forward to current day. Stored food in a plant. Emily, what do you call that? It's one of the three things you gave me the answer to just now. Stored food in a plant. One of the three things you said, not cellulose. Yes, starch, isn't it? Right? So the seed leaves themselves contain starch, but starch cannot be broken down to release energy directly. So we have to break down the starch first. And therefore, this is what we are looking at. What would an enzyme be? It is the same enzyme we have. It's going to be amylase. Once you can arrive here, it should be easy for us to choose D. Right? Amylase breaks down starch into maltose. Okay, so if there's no more starch, the iodine will not result in a change. Answer is D. YJ, there's a no, only the physical copies are available. We have lots available at all our centers, so please come on down and collect them if you want to. Okay, trypsin. So this reminds me of another question I saw as well. Trypsin, pepsin, erepsin, this one, I need to remember, okay, especially where they are from. Okay, pepsin is a protease in the stomach. Erepsin is a protease in the duodenum, the small intestine. Trypsin, if you're following the trend, is also a protease. I want you to tell me where is it produced by and where does it go to work at? Where does it function at? Produced by? Good answer, Brian and Joe. Unti, do you have an answer for this? Produced by the eating? Eating, do you know where is trypsin produced by? What produces trypsin? See? Pancreas. Thank you. Right? It's produced by the pancreas and released or secreted into the duodenum. Alright, so please keep this in mind. Release into the duodenum. Okay, so digest proteins found in milk. Okay, so here we're looking at the effect of temperature time taken for the mixture to turn colorless. So here we are already telling you that the color of the milk is due to the proteins there. When it turns colorless, it is because the proteins have been broken down. Similar to the previous experiment, where we have the cloudy solution in the test tube, after adding the gastric enzyme, the cloudy solution turns colorless. Here, we are looking at the time taken for the mixture to turn colorless. So here I want you all to recall the enzyme activity temperature graph and the enzyme activity pH graph. Which one is pH graph? Blue or green? Which one is pH graph? Hazel? Enzyme pH graph, blue or green? And therefore, the blue one will be the well done. Okay, this is very important. We need to be able to identify okay, so what does the red line? Dimson, can you help me to remind everybody the red line refers to the optimum. Right, so in the temperature graph is the optimum temperature, in the pH is the optimum pH. Thank you, Dimson. Right, so this is what we're looking at over here. Optimum temperature and pH is where my enzyme will function at its maximum rate. Okay, so if the enzyme is functioning at its maximum rate, it is working very quickly, right? If it works very quickly, will it take the longest or the shortest time to complete its job? Longest or shortest? 1K? Greatest rate of reaction, shortest time, right? And with that in mind, 1K still you. Answer should be C. Very good. Okay, we want to look for my optimum point over here. Answer is C. Nicole, we're still in discussions for that. Sorry for the delay. But if you're here now, then it's the same thing. Okay, so this is what we're looking at. Okay, moving on to our open-ended questions now, the free response questions. Like I said, we need to be able to identify from diagrams or drawings the different parts over here. Question tells us electron micrograph. Can I ask then, what's the biggest difference between an electron micrograph and a light micrograph? Okay, put it simpler. Light microscope and electron microscope. What's the biggest difference there? Anika? Light and electron microscope. Biggest difference is? Which one? You give me two things. Okay, so actually both are correct. But the first one, not necessary to know. Electron microscopes are humongous machines. They are literally the half the size of this classroom. Light microscope, you can hold in your hand, right? So there's a very big difference. Why? Electron microscope is much more sophisticated. It is unlikely that any one of you will ever use it unless you go into like PhD level. So I have never even used one before. Right? Why? It is a very sophisticated piece of equipment that uses a single beam of electron to shoot at the specimen to help you visualize those tiny little things. Okay, so that's the idea. That's the reason why electron microscopes can allow us to visualize such small cell structures. Okay, so the difference I was looking for is an electron microscope can allow us to see much smaller things in the cell. Okay, and the most obvious example we need to remember is the mitochondria and ribosomes. Okay, those two are very, very, very small things that can only be visualized with an electron microscope and not with a light microscope. Okay. Okay, so with that revision on the two different microscopes over here, we have a human sperm cell over here. We need to make sense of this picture. Correct now, we need to make sense of this picture. What is this picture going on? How is this even a sperm cell? Okay, usually we see the sperm cell drawing is correct, correct. Alright, okay, it is still this one. Let me tell you why. I take this sperm cell that we're all very familiar with. Is it? And then does it make sense now? Okay, right? So this is also a skill that we need to acquire, right? When you see these kind of questions in your own papers, I'm not going to be there to help you draw this, You need to be able to try and make sense of the diagram as best as you can. So now I have helped you try and make sense of this diagram. Can you then help me identify what A, B, and C are? Help me to identify what A, B, and C are. Keep it simple, Don't overcomplicate things. If the label point to a thing, okay, it is that thing. If the label points to a line, then it's just the line. The label points to the whole thing, then it's that whole thing. Myra, identify one of it first, please. Which one? Very good. B, A, and C. Which one? Spell, please. Well done for spelling the singular. Right, this one must be picky. Question label point to one. You give me the singular. Okay. And finally, C. Zixing. Excellent answer. Well done, ladies. Okay. Right. So this is what we're looking at over here. How did we know? Wait, how did you come to the conclusion that structure A is the mitochondria? It looks like one. Very good. So you have the description in your mind. But if no, this person never see before, how are you gonna tell the person that, oh, this thing, next time when you see this thing, it's mitochondria? What was used to describe the shape of the mitochondria? Sausage, like what? Sausage. You use this. Is this something you just thought of only? It's fine, right? Okay, we want to use more simpler shapes like circle, triangle, rectangle. Okay, so instead of sausage, we are looking at come, let's put this beside over here. I'm looking at three descriptors. Okay, rod shape. Sausage rod, kind of. Your teacher say sausage. I don't know whether it will be if they ask you describe the shape of mitochondria. Sausage. Wow, the teacher will see you hungry. Okay, so let's use simpler shapes. Circle, triangle, rectangle, rod, cylinder, even. Okay? Okay, wonderful. That's one down. Or you can even say oval, rod or oval shape. Okay? Okay, two more. Both have been mentioned in the chat already. Thank you. Come, ladies here. What are the two other descriptors used for the mitochondria? Come on, I hope you are not here to get answers. Please work your way a little bit, work with me, think about it. Oh, is it Reese? Reese says that the word sausage shaped is found in a textbook. Ah, goodness. The person that made the textbook must be hungry too. Okay, regardless. Moving on. How else can we see the textbook now? Like, I hope we don't see the sausage. Oh no. It is there. Is it? People in chat, they also checking. Is there an open inverted commas at least? Like sausage shaped.
It's just something shape.
I'm shooked. Okay, never mind. Rod shape or oval. We just stick to this. Okay, very simple. Yeah, write sausage at your own risk. Okay, moving on. Moving on. How else? What else do we use to describe the appearance of mitochondria? Thank you. Manifolds. Right? But where are the manifolds? Inside. So therefore, my second descriptor is yes. Thank you. Hey, I know that some of you have the answers in your mind on. Don't just sit down there, wait for me. Okay, please share this with us. We learn together as a class. Okay. So rod-shaped double membrane structure. Whether what thing has manifolds? Outer and inner membrane. Inner membrane has manifolds. Okay. All right, where the inner membrane is highly folded. Okay, these are our three descriptors. Three descriptors. And okay, so this is what we have to think of in talking about describing the mitochondria, right? Rod shape or general shape first. Then, oh, what is it made of? Two membranes means I must draw two lines. Outer membrane. But before I draw the inner membrane, I look at my number three. Inner membrane highly folded. Then. So roughly this can be a mitochondria. Okay, look at that. Uncanny resemblance. Okay, so this is our mitochondria. Alrighty, so with that in mind, mitochondria, why are there so many mitochondria in a human sperm cell? What's the function of the sperm cell? Am I right? What's the function of the sperm cell? Sorry? To go to the egg, what? To go shopping at the egg? What is the purpose? Okay, so it needs to reach the ovum for fertilization to occur, right? I want to focus on reaching or fertilizing when I answer this question. Reaching, very good. So what is the relevance of the mitochondria for reaching? Energy, I still need more things. What does the mitochondria do? Site of aerobic respiration. Thank you. Right, you need to have that site of aerobic respiration. Okay, so we're looking at lots of energy so that the part is the tail. What is the name of the tail? Starts with an F. Okay. What's the name of the tail? Thanks for trying. We're looking at the flagellum. Have you heard this word before? Have no. It's okay. So therefore, we are looking at let me add this in, please. To release a large amount of energy via very close, Ashley. Go try. Flagellum, is it? Flag. E-L-L-U-M. Okay, F-L-A-G-E-L-L-U-M. Oh, yeah, it's a okay, flagellum. Yeah, I mean you're not gonna be marked down if you say tail, just sharing with you what's the proper name of the part of the tail. What is that called? It's the flagellum. Okay. All right. And you notice I am using the word propel. Again, you're not going to get marked down if you say swim. That sounds weird to me. Okay. Simply put, interesting fact. How does the sperm cell move? When we think of a structure like this, how does it move? It actually doesn't move like a fish. It doesn't move like that. So if you look at my look at me over here, if my fist is the head of the sperm, my index finger is the flagellum. What it actually does is this action. It actually goes in a rotational action like this, right? So this is actually how sperm cells move. A pretty interesting thing that we refer to as a corkscrew action, right? Where it spins this way, rotates this way. Okay, awesome. Okay, let's move on. Where were we? Okay, so cross section of a dicotyledonous root here, right off the bat. What is a dicotyledonous? What does it mean to be dicotyledonous? Right, yeah, another episode of Bio Tongue Twisters, dicotyledonous. What is dicotyledonous? Anyone can tell me what dicotyledonous refers to? Which part of this humongous tongue twister can we break down into to make sense of? At least right? Do something. Okay, what is the something? Two cotyledons. What, therefore, is a cotyledon? Let me give you some examples. Green bean, dicotyledonous. Rice, monocotyledonous. Peanut, dicotyledonous. Barley, monocotyledonous. This one is referring to the seed itself. Refers to structures of the seed. Rice is monocot, green bean dicot, barley monocot, peanut dicot. So what is a cotyledon? Push yourself, think about answers, make mistakes here, we learn. That's the whole point. Mara, do you have a suggestion? No, it's not about growing where it grows, it's about how it has number of something. Dicotyledonous seeds have two, while monocotyledonous seeds have one seed. The seed have seed. By the time it's in the seed stage, there we don't talk about ovules already. Nope, not about transport vessels. Good try. What else? This one, bring yourself back down to primary school. Bring back down to primary school. Answer is a primary school thing. Green bean, dicot. Rice, monocot. Green bean got two what? The seed itself got two what? Layers? No. The green bean got two things. This is where the starch is stored at. Seed something. What? Seed leaf, yes, the seed leaf. The cotyledon is a seed leaf. Green bean, dicotyledonous, two seed leaves. Rice, one grain is one seed by itself, one seed leaf, monocotyledonous. Okay, so as far as your syllabus goes, you only need to know this. That's all. You don't need to know any more differences, right? So our syllabus, we are only focusing on dicotyledonous plants because the structures of the xylem and phloem actually are different. Since primary school, we learned that in a stem, the xylem and phloem are arranged in a neat ring around the stem, correct? This only applies to dicotyledonous plants. Monocotyledonous plants have no such neat arrangement. the xylem and phloem are scattered throughout the stem. Okay, so this is all you need to know, just very fleetingly, what's the difference between dicot and monocot. Make sure that we are aware that our syllabus focuses on dicotyledonous plankton. Okay, so therefore here, just remind ourselves very quickly, seed, leaves. Well done, Rachel and Joan as well. Okay, so here we are looking at a dicotyledonous root. I have filled in a table for you where structure R is the root itself. I would love for you to try and name P and Q and then write down the level of organization. When you see this word level of organization, what answers am I looking for? What answers am I looking for? What kind of answer? What word to put in these blanks over here? Level of organization. What are we looking for? Level of organization. Thank you. Wait, thank you. Whether or not it belongs to the level of a cell or a tissue. What's next? Organ. And then many organs come together to form the organ system. All the systems come together to form an organism. Thank you, Hui. So go ahead and try that. Identify P and Q and then write down the corresponding level of organization. Jenny, I think that's fine. Great answer, Kaden. Thank you. Sarah, are you looking for levels of organization? I did not put level of organization in the revision kit. Okay. Answer. Q phloem and then P is cell. Root hair and root hair cell different. Okay. Right. P is the root hair cell, but Q is not the phloem. Come. P is. Now, what is Q? Let me help you. Okay. Eyes on screen, please. What is Q? Very simple. See, magic. Xylem. Okay, xylem. Okay, sometimes it also appears as a three-sided thing. Also can, then it becomes the xylem still. Okay, so this is a silly little way to help you memorize more easily. How do I identify which one is the xylem, which one is the phloem? Okay, now functionally, how can I help you understand why this is the xylem? What is the main purpose of the root? Main purpose of the root, one word. What would that be? If I were to assign one word for the function of the root, what would that be? The word we're looking for is absorb. Xylem that transports. Okay, so if the root is the one that is absorbing water, it would make more sense that I need to have a larger xylem, because then all the water that I absorb can then be transported away by the xylem. Okay, so therefore I would have a relatively larger xylem compared to the phloem here. Okay, can? Also can, Ryan, if that helps. Okay, so here we have. Right, so level of organization. Obviously, root hair cell belongs to cell. Linton, you chose the xylem to be It's actually the next level. Tissue, okay. So this allows me to share with you a common question. So was it Brian?
Yeah.
Brian, right? Common question about level of organization. Define law. What does it mean?
I think got to go. So after that, from there, I go straight to Mummy's place.
Right. So defining the levels of organization. What is a tissue? Group of cells. Group of cells. They not just group. Then okay. Then I like call your tissue. Correct or not? Auntie, tissue is a group of cells. What? Group of cells working together to perform a similar function. You must have that. The idea is perform similar function. If not, why they group together? No point. Okay? So a tissue is a group of cells working together to perform a similar function. So if we elevate that, then the next level after tissue is organ. Organ. Sarah, explain to us in terms of the level of organization, what is an organ? No, in a similar fashion. Tissue is a group of cells working together to perform a similar function. An organ, therefore, is same. Same, exactly the same. Thank you. Right? So an organ is a group of tissues working together to form a similar function. So, question. Blood vessel. What level of organization? Blood vessel. What level of organization? Emily? Blood vessel is tissue, is it? Because it contains, well, everything contains cells in it. Blood vessel contains red blood cell. Okay, so always remember our definition for levels of organization goes according to the function. So when I ask you blood vessel, if you are to assign one word to the function of the blood vessel, what word would that be? One word for the function of the blood vessel. Thank you, Lim Zhen. Transport. Transport. Okay, so blood vessel is organ. It contains many tissues, the muscular tissues, the layers in the blood vessel. Okay? Awesome, right? Blood is a tissue, yes. Blood is a tissue comprising of plasma of your blood cells. Okay? Awesome, let's go. So, moving on. One of my favorite question types so that we can make sure we really get it right. Okay, so this one, what topics come to mind? Okay. Movement of substances, well done. Ying, which process comes to mind? If you look at this question over here. Osmosis, wonderful. So here we are tracking the red blood cell. Why is it so thin? We are tracking the red blood cell in distilled water. So therefore, we are looking at this one. Zooming in over here, I would like to remind us to use two descriptors for this current state. Anika, can I have one of them, please? Other word is? Keza? Just try.
Okay.
Expand. Okay, that is the word that I want to share with everybody to not use. Okay, things gain heat, expand. This one not about heat. Okay, thanks for trying. What other word are we using? it doesn't immediately burst, it must increase in size first, right? So that is the idea that Kaiser had in mind. Well, increase in size, expand. Like expansion is only for gaining heat. Okay? Thank you for trying that, right? So it swells and then eventually bursts. We can also use another word here. It starts with letter L. Must be truthful. Cannot tell lies. Okay, right? So you can either say it swells and bursts or swells and lyses. Okay, answer goes lyses. Nicole's answer is yes. Okay, so like I shared at the start of lesson here, the most recurring topic and concept that we are looking at here is movement of substances, because very simply put, like what N shared with us earlier on, whenever we are explaining MOS questions, 80% of the answer comes from the definition. 80% of the answer comes from the definition. Where is the other 20%? Where is the other 20%? 80 comes from the definition, the 20 comes from answering the question itself. That's it. Your explanation is your definition very vague, right? You need to tailor it to answer the question specifically. The additional 20% that this question is requiring is the appearance portion. Your opening sentences is just osmosis. That's it. That's 80% done. The remaining portion, explain the question. Question say, appearance. Oh, talk about the appearance. Why appearance like this? Okay, can? All right, so over here, we are looking at the red blood cell swells and bursts. Why? So here, Yining mentioned osmosis. What is the name of the gradient here? What is the name of the gradient in osmosis? Water potential. Thank you. Water potential gradient. Okay. So if I have the red blood cell swell and lyse, the gradient, is it into or out of the cell? Into or out of the cell? Into. Very simple. Okay, listen to this phrase first. Water enters the cell, causing it to swell and burst. Incorrect. Why? Let's see louder, please. Water molecules enter the cell, causing it to swell and burst. Still incorrect. Kaiser, what are we missing? Okay, not the. I'm talking about the idea of not the specific things here. You're correct. That one needed, definitely. Okay. Water enter, cell burst. Too much water. Thank you. Who was that? Was you, Clarissa? Thank you. Too much water. Water is constantly entering cells, constantly exiting cells. You must have the idea in mind. So water always enter, water always exit. When then will it burst? Too much water. To make it easier for you, instead of too much water, just say excess. Okay? Okay, very straightforward. The first marking point is always about which one got higher and lower water potential. Okay? So let's write it down together. Okay, distilled water. We start distilled water. We put it out higher so that our friends at the back can see better. Fill in the blank for me, please. Thank you, Linton. Correct. Well done. Resulting in a net movement of water molecules. Zing. Across. Osmosis can only occur across A. Very good. I also want to be specific. I remember the 20%. So not just partially impermeable membrane, partially permeable cell membrane. Okay? So we have achieved our 80%. What's the question asking us again? Very good. The appearance. So final 20%, let's talk about the appearance. Like I said, water in, water out is very normal, very common. In fact, required. So here, what must we talk about? The water. Excess, excess. Okay? All right, I'm seeing some good habits over here. I'd like to encourage everybody. I say this to all my students over here. After copying down something the teacher has said, after writing down, take a moment to read what you have written yourself on your own paper, not on my screen. Right? What you've done just now is very passive. Copying, write down. Now I want you to look at what you have written and understand your own writing. Let me give you a minute also to do that now. And if we are ready, please get your inner artist caps on and pencils out, please. I always share, no matter what level of education we are at, whenever we are asked to draw something, pencils out, please. Question is asking for us to draw what the onion epidermal cell will look like after being placed in a concentrated solution for 1 hour. Ignore the dots, just draw whatever thing will be necessary. Alright, let's go ahead and try that. Yes, Brendan, well done. This is also another reason why we take lessons. Then I can tell you what all these weird little things are. And after today, not weird anymore. Okay, so let's have a look. Let me show you a drawing. I'm very honest. I'm not going to use my snap to the picture on my iPad. Okay. Did we draw this? Okay, firstly, if you drew this, it is still incorrect. We are missing one thing. Firstly, can we try to understand first? When I put it into a concentrated sucrose solution, what is the movement of water? Into or out of the cell? Out of the cell. Thank you, Myra. Right? So when there's an excess exit of water out of the plant cell, what are the two descriptors we need over here to describe the state of the cell right now? What shrinks? We need to point to a specific cell part. Vacuole, thank you. Right, so yeah, just very quickly, vacuole shrinks. Second descriptor is the thing that makes this diagram weird. What's the second thing? Usually we draw them together. Now they are separate. Yes. Thank you, Myra. Woo, I went to my pen. And therefore, we have found our mistake in this diagram. This one cell membrane, right? Then okay, what? Pulls away from the cell wall. What? The problem is the cell wall is not a line. The cell wall is a layer. Okay, so if I'm going to these parts so that I can help you see the difference over here. Okay, here, why must draw 3 lines? When this diagram, if I were to draw it, I will only draw 2 lines. Simply because, come, eyes on screen, please. If I were to draw cell wall, and then I'll draw cell membrane. So when we draw it, obviously there are only 2 lines. Because in that state, the cell membrane is fully attached to, fully adhered to the cell wall. Because the cytoplasm is full of water. It is pushing against the cell wall entirely. So therefore, cell membrane line, cell wall line, draw as 1 only. But when excess water exits the cell, there's less cytoplasm. So the cell membrane now pulls away from the cell wall. This leaves behind the entire 2-lined structure of the cell wall. So it's very important for us to be able to correctly label the space as the cell wall and the inner line as the cell membrane. Is this clearer now? Please make sure we have this correct. Any questions to ask so far? No questions? Then I ask questions. What is here? What is in this region? I heard the answer. What was that? Say it again. What is it? Why is it a sucrose solution? Come, Auntie, why would this region be filled up by the sucrose solution? Hold that thought. Okay, so I have 2 answers that I want to share. Again, not naming and shaming. It's about learning properly. Cannot be vacuum. There cannot be nothingness there. Okay. Number 2, cannot be air space as well. If there was no air, there was no vacuum, it cannot just appear by itself. Can try to understand that first. Very common answers as well. So get that out of the way. Okay. For those who supplied those wrong answers, thank you for allowing me to share with the class here. Okay. Right. Sucrose solution cannot enter the cell membrane. I shared with you earlier on. Sucrose cannot pass through cell membrane. Okay, so why can it pass through the cell wall then? We are thinking of the property of the cell wall. What of the cell wall can allow the sucrose solution to enter, but the cell membrane does not allow? What property comes to mind? The cell wall is fully permeable. Thank you. The cell wall is fully permeable. So therefore, whatever is outside will also be found inside the cell wall. But the moment you get to the cell membrane, different story applies because the cell membrane is partially or selectively permeable. Okay? Okay, so with that, these are our two descriptors. I'll leave my screen on this answer here because I would like for you to try and answer B part 2. B part 2 asks us to explain why that plant cell became like that. There's a reason why I did this question with you earlier on. There's a reason why I'm leaving it here for you as well. But a word of caution, don't happy, look, copy, then say water enter also. There will be people doing that. Water exit. Okay, can you go ahead and try that now, please? Make sure we have fully absorbed, pun intended, this format of answering here. Okay, and as you are trying that, just know that this is not the only way to answer. It is not the absolute best way to answer. It is my suggested best way to answer. Okay, find what works for you. Please go ahead and try that now. Wait. Okay. Brian, I need to show the answer. That's the point. Okay, I'm going to zoom in into the answer. What's it doing? Pass the pause, cell membrane, yes. So it doesn't move. So then what moves? Other than sucrose, what is called sucrose solution? Okay, so the blue and the green portion, very simple, it's just direct split. You can refer to the cell cytoplasm having a higher water potential than concentrated sucrose solution. Or you can flip it around and say that the concentrated sucrose solution has a lower water potential than the cell cytoplasm. Either way works. Okay, you just need to be able to tell me that there's a net movement blah blah out of the cell. Everything after the cell will be the same. Then when you look at the orange one, instead of excess entry of water, it will be excess exit of water. Okay, like I said, you don't have to go word for word. It is the content that matters, the idea, the message that matters. Over here, I'm just going to say, hey, the excess exit of water molecules out of the cell. Then this is where I point out two descriptors. Causes, number one, vacuole shrink. Number two, cell membrane to pull away from the cell wall. Okay, so the wording is pretty important over here. Shrinks from cell wall. Okay, so we take the words together. Shrink to vacuole, pull away to cell membrane. Okay. Any questions on this? Like I said, I want to emphasize for MOS, follow your definition 80%, other 20% answer the question itself. Alright, so at the end of the two sessions, the answer key will be given to you. So if you cannot follow too much here, I only have a few minutes left. Allow me to move on. Okay. Alright, here we have a very short question. So this one, I will just walk you through it very quickly. I'm just going to highlight a few things over here. We are looking at oil stains. What will oil contain? What biological molecule is oil? Fats, very good. Thank you. Okay, fats. You can also use lipids, whichever you prefer. Okay, next up, I have biological washing powder. What would the washing powder contain that would allow me to say it is biological washing powder? Keep it in mind, I'm using it to wash away oil stains. And this thing is also sensitive to temperatures. What would this biological thing could be? Enzymes, absolutely. With that in mind, 15, 40, 75. At which temperature would I therefore have the oil stain become the clearest? What temperature would that be? 15, 40, or 75? Likely to be 40. Why do we choose 40? It's closest to what? To body temperature. Very good. So when we talk about thinking about biological, we think about enzymes. If the question doesn't state it, go to body. And go to body temperature. So therefore, this one will be our cleanest at 40 degrees Celsius. The middle one, no change at all. The last one, little bit of change. Which one do we go for? The 15 or the 75? 15 is the darkest one or the light one? Second or third? Okay, sorry. Second is 15 or 75? This one, 15 or 75? Why 75? Very good. Very important. Above optimum, denature. Denature, D for denature, D for die. Enzyme cannot function already. So it remains stained. If above optimum is denature, below is, or what is that? Inactive, thank you. Inactive means you're sleepy, but you still can do a bit of work. So you're still going to be able to break down the oil, but not as effectively. So therefore, I'll say it's 40, 75, 15. Okay. Right, the main component of the biological washing powder to remove the oil stain, we already talked about it. You can either write down the word enzyme or the specific enzyme. What would that be? What is the specific enzyme that breaks down our fats or lipids? Lipase, very good. Okay, so either enzyme or lipase. This one fairly straightforward. If I wash 90, do we agree with Eunice's statement here? Cannot, what will happen? We just talked about this, right? If it's above optimal, what happens to the enzymes? Denature. Okay, so this one just put it down at the points at the side here, right? I would like for you all to tell me the answer of part C. Tell me what is the test you will conduct first. What test will I conduct? Would I choose to describe part C? Can I just write down the name of the test on your paper, please? Can I write down the name of the test on your paper, please? Hey, very good. I'd like to give myself a pat on the back as well because everybody chose ethanol emulsion, right? Well done. Everybody chose the wrong answer together. Read the question properly. Describe the test for the substance in B part 1. What do we put in B part 1? Enzyme, right? What are enzymes? Yes, proteins. Test for the substance in B part 1, which means test for lipase. Lipase, enzyme. Enzymes are proteins. Hence, this is the Buret's test. Okay, right. See everybody get turned around, okay? Buret's test for protein, So please make sure we are understanding the question properly. Anyone still not convinced it's Buret's test? Okay? Okay? Awesome. Let's go on. Okay, thanks everyone at home for getting tricked as well. This means I did the question correctly. Okay, so moving on. Final question, right? Give me a couple more minutes. We are looking at 4 flasks, P, Q, R, and S. We are looking at the different conditions I am putting in my starch solution to. The first one, P and Q. 5 cm cube of saliva versus 2 cm cube of saliva. What is the factor that I'm studying here? What factor am I studying here? The effect of Sarah? What about the enzyme? Both also have the enzyme, right? So therefore, this one is the effect of enzyme concentration. Can? In flask R, what does boiling the saliva do? We talked about this many times today. Boiling the saliva results in Kaiser? Thank you. Exactly what I was hoping to hear. And in flask S, which is similar to flask Q, however, we have 10 degrees Celsius, and therefore the word we are looking for over here is 1K. Okay, so what are we looking for? 10 degrees Celsius, very cold, below optimal. Enzymes are, what is that? Inactive, thank you. And whatever that was before as well. Okay. The copy is the one that I forgot to take out the answer one. Right? Is it printed for you already? Okay, so if you look at the answers over there, does it make sense now? In R, salivary amylase is entirely denatured, so no starch can be broken down. That's why curve 4. Make sense? Okay. In flask P, highest concentration of enzyme, greatest rate of reaction, starch concentration reaches 0 the fastest. Therefore, curve 1. Make sense? Okay, so that's the explanation there. And fairly straightforward for part B. To wrap off today, which curve will exactly match the result for flask T? In this entire paragraph, you actually need to only jump to one word to find the answer. Hui, what allowed you to find the answer so quickly? Very good. Anything along the line of the acid. You look at the word acidified, or you look at the entire hydrochloric acid portion. What happens to the enzyme when they are put into very extreme pHs? Denature, thank you. And that is the word we wrote down earlier on as well. So if in flask R, we also have denaturation, you will also therefore have the curve 4. Wonderful. Okay. All right, like I said, explanation is what I just said. It will be provided in your answer key as well. And that wraps up our lesson for today. Okay, so right, so just a closing one here. This is session 1 only. That's what we are looking at for this question, for this session over here, just those 3 topics. Next session, can I assume that I'll see everybody again? Anybody not coming on the next session? You're not coming? Why? Never get or not relevant to you? Who are you? You're gonna come online then? You're coming? Okay, the thing is, okay, wait. Okay, yeah, so friends at home, I hope you have enjoyed that as well. If you are going to attend the second session, I'll see you online as well. Next session is when? Yeah, I also forget. Let me check. Is it tomorrow? Oh, tomorrow. 4 to 6, correct? For tomorrow 4 to 6. Tomorrow 4 to 6 is the next session. Okay. For those who have gotten the physical tickets, it is still here as well. So let me let the people at home go first. I hope you have found some use in today's session. If not, enjoy the rest of the September holidays. Wait, wait.